presentation, Dr. Schiller. So as we heard, CAR T therapy has changed the way we think about cancer treatment. However, the process is complex. As you can see here, you start with an impaired starting material. There's complex logistics. Time to delivery to the patient is lengthy. The cost is high. And the limited availability creates a challenge for multi-dosing. And at the same time, when you engineer the product, not every cell is engineered, and not every engineered cell is pristine. At Fate Therapeutics, we've been working on using IPSCs as a starting material. These are pluripotent cells that are maintained in a petri dish, have the unlimited self-renewability, and can at any time become any of the 200 cell types in the body. We've taken advantage of this product, using it at a single cell level to do precise engineering, to create master cell banks, to create uniform composition of cell material, and have extensive characterization to be able to create clonal cell lines to make homogeneous cell products. This is done by the scheme you see here. We reprogram, engineer, and we drop single cells into a 96-well plate, allow that population to expand. It becomes a continuous culture. The culture maintains its pluripotency, genomic stability, and now you have the ability to do very precise engineering. As you can see in the middle, you can drop several genes into a targeted locus and end up with a pure population of cells carrying those genes. In another strategy, you can knock out various genes. Here, we targeted beta-2 microglobulin in a biallelic manner at the single cell level, got rid of the expression of B2M, and hence HLA class 1 expression. So now we have a pure product that is knocked out. We take that engineered product and we induce differentiation into hematopoiesis. So here we go from an iPSC to a CD34 positive hematopoietic progenitor cell and continue that differentiation into NK cells and T cells. Here we're illustrating NK cells. You can see that nicely the population becomes homogeneous for CD56 positive, which is a pan marker for NK cells. Other activating receptors on the NK cells also come up, as you can see here, NKP30. The process is highly scalable. 1 million iPSCs gives you 1 E12 NK cells, and that output is continuous. You can every single time start this process and deliver 1 E12 NK cells. At the global gene expression level, these NK cells look very similar to their primary counterpart, and this process has been transferred to now manufacturing process, where it's done in multiple sites. Our collaborators at University of Minnesota and internally at Fate. The product is released based on the fact that it's pure for hematopoietic cells and NK cells, has high viability, and now allows you to have a homogeneous, high-quality product that's low cost. Each dose is $2,500. It's thawed and directly infused. There is no processing needed, so it becomes a true administered off-the-shelf product in an outpatient setting, and now is analogous to pharmaceutical drug product development. This process, as I mentioned, starts with an iPSC population. It's engineered. Single cells are selected. A master cell bank is fully characterized and allows for production of T and NK cells in an off-the-shelf manner. Using this process, we have now started 3 clinical investigations. First is FT500, which is the first iPSC-derived product cleared for clinical investigation in the US. 12 patients have been dosed. Combined 60 doses have been given to those 12 patients, and so far the product has been safe and well tolerated. FT596 is the first engineered product. It's the first engineered iPSC product in clinical investigation in the world, to our best of our belief. And first patient with the lowest dose has shown anti-leukemia effect and also persistence in the bone marrow. FT596 is a multi-factor engineered product, which I will talk about. FT596 consists of 3 anti-tumor modalities. It's the first product, first cancer immunotherapy product to be engineered with 3 active anti-tumor modalities. It consists of HNCD16 and high affinity non-cleavable CD16, which binds to antibodies and elicits antibody-dependent cellular cytotoxicity. This is a key action of general killing by NK cells, and we've enhanced that feature. So now we can combine FT596 with any monoclonal antibody to elicit enhanced ADCC. We've also introduced a CAR that's been calibrated to NK biology, targeting the CD19 antigen. And finally, we've added IL-15 receptor fusion to support persistence and reduce the dependency for exogenous cytokine support. Because the clone was selected to have one copy of each product, you can see on the far right-hand side, the top should say CD16, the orange should say CAR19, and the bottom should say IL-15 receptor fusion. You can see that each are expressed homogeneously, and this is very challenging if you want to engineer NK cells or even T cells to have this profile. So compared to primary NK cells, you can see such uniform differential. To show what step manner these engineering attributes do, we created a Coculture assay, which consists of cancer cells that are CD19 positive, CD20 positive. So these are antigens on B cells that are usually targeted, as we heard earlier. We also co-cultured with CD19 negative, CD20 positive cancer cell line. When we introduced the CAR19, you can see you can effectively eliminate the CD19 positive target cells, and when you add the IL-15, it's completely eliminated because IL-15 enhanced persistence and durability of the product. With utilizing the agent CD16, we add rituximab, the anti-CD20 antibody, and clear the second half of the product that you could argue mimics antigen escape. We use CD20 here. We could have used CD22, CD123, or other monoclonal antibodies that target B-cell malignancies. This product in vivo is highly efficacious as a monotherapy targeting leukemia. It has the ability to control tumor in an extensive period. In combination, going after lymphoma, you can see where rituximab fails to control the tumor burden. In combination with FT596, it does a great job maintaining durability and control of the tumor. And finally, in an allogeneic system, you can see that compared to CAR-T, FT596 as a monotherapy is comparable, if not better. So my final data slide, I will go recap what this combinational strategy is. So here we used Raji lymphoma cell line, that's CD19 positive, CD20 positive, and compared FT596 to 5 different primary CAR-T products. As you can see here in this low effector target ratio, so in vitro stress test, where only 0.3 to 1 ratio of the effector cell is compared to the target. You can see that FT596 is comparable to primary CAR-T. However, when you add Rituxan, you can see that you get a more deeper durable response. In a different format, trying to mimic antigen escape, we knocked out CD19 from the Raji cell line and created CD19 negative, CD20 positive cells, and then introduced a high capacity test to look at effector function. As you can see here, primary CAR-T and FT596 alone failed to control the tumor elimination over a long-term period. However, in combination with Rituxan, FT596 was able to eliminate the tumor load in the vitro culture. So this is what's being presented at ASH for Fate Therapeutics specific to CAR programs. As I mentioned, FT596 is publication number 301, presented later today. We also have a CAR BCMA in combination with Darzalex product where we've knocked out CD38 to eliminate fracture sites. So this is a true NK product that can sustain ADCC as well as CAR BCMA activity. And this is publication number 3214, IND to be filed second half of next year. And excitingly, we are also working on first off-the-shelf CAR-T product where we're utilizing some of the top engineering attributes that's coming out of Sloan Kettering and out of Dr. Michelle Satterline's lab to create a first off-the-shelf CAR-T product, and they'll be filed in a few months. And that's publication number 4434. Thank you.